Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 44, and it's going out on January the 30th. It's a 5-Friday month, and I thought that was maybe rare, but there's another one in May and another in October, so maybe not that rare. I thought my voice was back to normal this week until I started chatting to someone while hiking and realised that there was hardly any volume, so clearly the struggle continues. It has been wild and windy here, but certainly better than the blizzards that they've been having in parts of the US and Canada. Having lived in Canada for many years, I love how the news reports we get here in the UK talk about massive storms in the northeast of the US, as if the border control in Canada just turns the storms away. Last week we had a conversation with Lisa Garetas, Executive Director, Pharmacology and Translational Science at Artiva Biotherapeutics, and Avacta Therapeutics CEO Christina Coughlin. This week we turn our attention to breast cancer with an interview with Pan-Cancer T CEO Dr. Rachel Abbott, and we also have an interesting interview that considers the big picture of drug development as we not only talk to Erik Manting, CEO of the immunotherapy company Mendus, but also Janet Hoogstraate, CEO of the CDMO helping them get where they want to be, X Biologics. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us or sponsoring a podcast episode. And it's also the same address if you are looking for event coverage. Alright, let's take a look at the news that you may have missed over the past week at deeptechdigest.com. Zaire Ebola virus vaccine development is moving forward. We had an article on how cancer cells protect themselves from the immune system, and Lilly's platinum-resistant ovarian cancer drug has received US FDA breakthrough therapy designation. The group Women in Life Science Denmark, or WILD, has expanded with the launch of WILD Finland. Infinitopes has raised more funds for its precision cancer vaccine. Nanoscope is boosting retina gene therapy in Japan. DIA has launched a platform to boost drug development. And there was an article on why tumours become aggressive and genetically unstable. There are new cancer treatment options through expanding an ADC payload spectrum. Resolve M Therapeutics has emerged from stealth, and two-faced nanoparticles help antibiotics treat resistant bacteria. And by two-faced, it doesn't mean that they say one thing to your face and another behind your back. Anyway, you can read all of these and plenty more at deeptechdigest.com. Alright, on we go, and let's have the first conversation. And we found out all about T-cell receptor or TCRT therapies for hard-to-treat solid tumours, including triple-negative breast cancer, with the CEO of Pan-Cancer T, Rachel Abbott. Hi Rachel, thanks so much for joining us today. Looking forward to learning a lot as usual. So to start, maybe you could give us a bit of background on the company, Pan-Cancer T.
Absolutely. So Pan-Cancer T was founded in 2020. And it's a spin-out from Professor Reino de Bet's lab at Erasmus Medical Center in Rotterdam in the Netherlands. We built on more than 20 years of pioneering research in adoptive T-cell therapies. Our mission is simple but ambitious. We want to empower a patient's own immune cells to tackle hard-to-treat cancers, especially solid tumours. From day one, we focused on overcoming the two biggest hurdles in this space. One, finding truly tumour-specific targets, and 2, making T cells resilient in the hostile tumour microenvironment.
All right, so could you expand a little on what TCR-T cell therapy is and how it works?
Certainly. So adoptive TCR-T cell therapy uses a type of white blood cell, the T cell, that normally recognizes and kills virus-infected cells. And the therapy involves taking a patient's own T cells and genetically modifying them so that they express a T cell receptor, what we call a TCR, that enables the T cells to recognize a tumor-specific antigen. We then expand these cells and infuse them back into the patient so that these engineered T cells can seek out and destroy cancer cells that display that antigen.
And is this an area that many companies are working on?
Yes, this field is one that is growing quickly. So companies like Adaptimmune and Immatics have really led the way, and they've focused on targets such as MAGE-A4 and PRAME. And there are others, including Anoka and T-Knife and Captain T-Cell, that are also active. However, most programs are limited to rare cancers, or they focus on the same small set of targets that are generally expressed in a small subset of patients. PanCancer T is different because we focus on novel tumor-restricted targets that maximize the likelihood of responses in indications with high unmet need. So, for example, ROPN1, which is the target for our lead program, PCT1 costim, is strongly and homogeneously expressed in over 90% of patients with triple-negative breast cancer, TNBC, and melanoma. In addition, we also combine this with proprietary next-generation engineering, which is designed to improve T-cell durability, which we aim to transform the outcomes where current therapies are falling short.
And what are the differences between TCR-T cell therapy and CAR-T cell therapy?
Yes, so CAR T cell therapies use chimeric antigen receptors that are based on antibodies, and these can only target antigens that are on the cell surface. And that represents really only a small fraction of the proteome, and those antigens are often shared with healthy tissue. By contrast, TCR-T cell therapy use T cell receptors, and they can recognize peptides that are derived from intracellular proteins that are then presented on the cell surface by HLA molecules. That's really a game changer for solid tumors in particular, where good surface targets are scarce.
And so what kind of cancers would it be applicable to?
So our platform can address multiple solid tumors, and those could include breast and melanoma, colorectal, ovarian, and more. We have chosen triple-negative breast cancer, or TNBC, as our lead indication because it's the most aggressive subtype of breast cancer with very limited treatment options and a poor prognosis. So median survival after metastasis is less than 6 months. And our ROPN1, our lead target, is expressed in over 90% of TNBC cases, making it a really ideal starting point for helping patients with this particularly aggressive tumour.
I wonder if you could tell me what is special or different about TNBC as opposed to other breast cancers?
TNBC is a particularly bad form of breast cancer. I think a major part of that is that triple negative means it's lacking the overexpression of estrogen, progesterone, and HER2. Now, all the targeted therapies that have actually transformed breast cancer care target those. And so fundamentally, the treatment options for triple negative breast cancer are massively limited. So I think that is largely the problem. And people haven't tried treating with T-cell therapies. So I guess the point I was trying to make is that actually because they are younger and they've not gone through many forms of treatment, actually they might respond reasonably well to a T-cell therapy. And what's really interesting, so ROPN1, our target, is found in triple negative breast cancer, and it seems to be in almost all forms of triple negative breast cancer, and yet we don't see it in other forms of breast cancer, which is intriguing because actually there aren't generally any markers that define triple negative breast cancer. It's just you lack all the other things.
And so how are you overcoming the challenge of keeping T cells functional after reaching a tumor?
Oh, that's a great question, and really has been the focus of our next generation technology development. So solid tumors create a hostile tumor microenvironment that suppresses T-cell activity. Our proprietary co-stim technology addresses this. We embed a co-stimulatory signal actually within the TCR itself. And when the engineered T cell encounters its target, it will get an activation boost, even in the absence of natural co-stimulatory ligands. Those ligands are generally absent in tumours. And this approach really improves T cell fitness, persistence, and phenotype in our preclinical models. And we really believe that it can add benefit when we go to treat patients.
The first drug that has been approved in this space, Tecelra, it's had several side effects, including the possibility of cytokine release syndrome. How are you addressing issues like that?
Obviously, safety is paramount, and we've done an extensive package of preclinical studies. And really, they show that, amongst other things, that PCT1 co-stim does not recognize healthy cells, and it doesn't trigger aberrant cytokine release in predictive assays. So we hope that there will be a limited problem. But on top of that, our trial design includes sentinel dosing and close monitoring to mitigate risks like cytokine release syndrome. I think it's also worth saying that I think there is a bit of a balance. So actually, from all the kind of adoptive T cell studies that have been done, I think there's probably an acceptance that some level of cytokine release is a good sign. And actually, you need cytokines to be produced if the T cells are going to be attacking the tumour. So there's definitely a balance that you want to kind of hit, and hopefully we will do that.
Right. Have there been any other challenges along the way?
I would say we've been surprisingly fortunate with this. I mean, I think we have built on a lot of experience that has been developed both by us within the lab that the company spun out of, in other companies that I and others in the company now have worked at. And we've really kind of, been able to utilize all of that to make sure that we ask the right questions, that we have chosen, when we chose our lead TCR, we had a choice. And we have kind of called upon what's happened in the past to enable us to make that decision. And I have to say that actually everything has gone almost surprisingly smoothly. I really hope that continues in the future.
Definitely. Could you tell me a bit about your clinical trial pathway and the recent funding announcement?
Yes, so we've just secured 10 million euros. So that includes 5 million from our existing investors. And then we've also got a 5 million innovation credit loan from the Dutch government. So that's really an instrument that the government have put in place to support companies like ours. And overall, this money will enable us to take our lead program, PCT1 co-stim, into a first-in-human trial for TNBC. We plan to file our clinical trial application with the EMA in the middle of this year, 2026, and hope to start treating patients in early 2027. The trial will assess safety, tolerability, and early signs of efficacy in late-stage metastatic TNBC patients at a few sites within the Netherlands.
What is going to represent success for you with those trials?
Our ultimate goal is fundamentally to change outcomes for patients with TNBC and ideally demonstrate curative potential along the lines of that seen with CAR T cell therapies within white blood cancers. But ultimately, for the first in-human trial, success means demonstrating safety and early signs of efficacy, such as tumour regression or durable disease control. Longer term, we aim to progress from second-line to first-line treatment and establish TCR-T as a standard of care, and hopefully expand into additional indications where our technology can deliver transformative benefits, such as in melanoma.
And do you think that long-term that you can make breast cancer something that's manageable or even cure?
Yeah, so, I mean, we'll have to wait and see. That would be our hope. Certainly, as I said, some blood cancers have been cured with adoptive T cell therapy. Solid cancers are without a doubt more challenging, but we are really trying to develop a T cell product that is likely to work better in solid cancers. So with our co-stimulatory technology, but also in terms of the choice of the target, it really does seem to be strongly and homogeneously expressed in the vast majority of tumours. And we do know that there's good correlations between antigen expression levels and response rates. So it looks promising. I think also the patient population is one that might be more amenable to good T cell responses. On the whole, TNBC patients tend to be relatively young, and they haven't gone through many, many rounds of other prior therapies because there are so few treatment options. And because we're using the patient's own T cells, we want people who have pretty fit T cells, and both age and lots of standard care for cancer tend to have an adverse effect on T cells. So hopefully our patients will have a pretty good starting point.
What other kinds of treatments are being developed for this disease?
So at the moment, current options include chemotherapy, immune checkpoint inhibitors, PARP inhibitors, and antibody drug conjugates like sacituzumab govitecan. And while these offer incremental benefits, progression-free survival really remains limited, and I know toxicity is a very significant concern. We've got feedback from key clinical opinion leaders, and that has really underscored that there are very few promising alternatives on the horizon. So there's a clear need for transformative approaches like TCR T-cell therapies. And yeah, the clinicians we've spoken to are all very excited about being involved in the trial.
Do you anticipate that this, your treatment will become part of a combination of treatments?
In the initial clinical trial, we'll really evaluate PCT1 co-stim as a monotherapy. Looking ahead, we could definitely consider combination strategies, particularly with immune checkpoint inhibitors or antibody drug conjugates. That might allow us to maximize therapeutic impact, and it also potentially could enable us to treat patients earlier in their care pathway as well.
Are you currently looking for partnerships?
Partnerships are indeed central to our strategy. So for PCT1 co-stim, we ultimately aim to collaborate with a pharmaceutical company that will enable us to complete clinical validation and then execute all the many activities that would be required for a market launch. We're really hoping that the clinical proof of concept data that we'll generate with our current funding will be pivotal to enable those kind of partnerships. Additionally, because our technology is modular and co-stim can enhance other TCR T-cell therapies, we are hoping to be able to form partnerships that are focused on integrating TCR co-stim with other TCR platforms. And then beyond oncology, which has been our focus, we see potential applications in autoimmune indications, and that would further expand the reach of our innovation, but we probably would want to do that in partnership.
All right. Is there anything else in the pipeline?
Yeah, we have other pipeline projects. We've got another TCR that we're interested in developing further for a range of other tumours, including colorectal. We're also developing other next generation approaches as well.
So where do you see this field of TCR T-cell therapy heading?
Yeah, so the approval of Tecelra in August 2024 as the first TCR T-cell therapy really was a watershed moment. And over the next decade, we really expect TCR-T to expand as a key modality for solid tumours, and hopefully driven by innovations like ours that improve durability and safety. Currently, obviously, TCR T-cell therapies are autologous. That is, they use a patient's own T cells. And that process presents significant challenges. At Pancreas T, we've really invested heavily in developing a robust, cost-effective autologous manufacturing process that we believe is essential for developing a successful T-cell product. However, looking forward, the field will likely shift to in vivo delivery, especially if the promising early stage clinical data that is behind a lot of the major M&A deals that we saw last year continues to gain traction. And actually, we're actively exploring partnerships with in vivo delivery companies with a view to developing an in vivo version of PCT1 co-stim, which could ultimately be positioned as a follow-up product once we've validated our novel target and receptor with our autologous version. And then in addition to that, our next generation TCR co-stim technology, we think potentially offers enhanced armoring and expansion advantages for in vivo TCR T cells. And it's got a pretty small payload, which would be compatible with the viral vectors that people are using for these in vivo delivery platforms. So we can see potential sub-licensing opportunities for integration with other TCR platforms.
And seeing as we're at the beginning of a new year, how do you see 2026 for PCT and beyond, I guess?
Yeah, so I mean, in the near term this year, we are finishing our CMC work in the next month or a couple of months. and then looking to complete our CTA package and file that in the middle of the year, and then really initiate the first in-human trial for PCT1 co-stim. So we'll aim to start treating patients this time next year, and we're obviously really excited about that. Beyond that, we'll be expanding our pipeline to other solid tumours. We're obviously looking at exploring partnerships and continuing development of next generation technologies that make cell therapy more effective and accessible.
So now, if you didn't already, hopefully you know a little bit more about TCR-T therapies and triple-negative breast cancer. Now, we often have drug development companies on the podcast, and today's no different. And also, occasionally, we have CDMOs and CROs as well. Today we have a conversation that kind of completes the circle in a way. Mendus is a Swedish-Dutch immuno-oncology company developing cell-based therapies aimed at stopping cancers from coming back, rather than treating only the initial tumour burden. Mendus is collaborating with CDMO NorthX Biologics, and the two companies have worked together since 2023 to establish scalable manufacturing for vididencel. In late 2025, they announced successful GMP manufacturing certification for vididencel at NorthX's facilities. And so we'll talk about all of those things with Mendus CEO Erik Manting, and first with NorthX CEO Janet Hoogstraate. Hi Janet and Erik, thanks so much for being with us for the podcast this week. Maybe first we could have an introduction to your respective companies, and maybe we'll start with NorthX.
So my name is Janet Hoogstraate. I'm CEO of NorthX Biologics. NorthX Biologics is a CDMO and an industrial infrastructure in Sweden for advanced biologics. We've been around as NorthX for 4 years now, but we build upon extensive GMP experience in pharmaceutical manufacturing in Sweden. Our headquarters is in Matfors in the middle of Sweden, close to Sundsvall, and we have a second site in Stockholm. The company works on advanced biologics. It spans all the way from microbial and mammalian proteins, including antibodies, fusion proteins, but also more in the advanced therapies, so gene and cell therapies.
Yeah, my name is Erik Manting. I'm CEO of Mendus, a company based in the Netherlands and in Stockholm, in Sweden. We have discovered a way to stimulate the immune system so that it can recognize what's called leukemic cells. Leukemic cells are cancer cells. They cause blood-borne malignancies, particularly acute myeloid leukemia or chronic myeloid leukemia, which are in principle deadly diseases. So you have to treat them or continue to treat them unless you find a way for the immune system to take control and basically make sure that the cancer cells can no longer grow out to form blood-borne tumors. And that can be done with immunotherapy. And that's a concept we have been working on. We've also developed a production process for our product called vididencel, which is a whole cell-based immunotherapy. And we have also, of course, tried to look for a way to make the cells at large scale. And that is why we teamed up with NorthX Biologics in 2023. And in the meantime, we have established in that collaboration the large-scale production of our product vididencel, so that we can now actually move into late-stage clinical development and in the end commercialization of the product.
All right, very good. Could you tell me a bit about how the partnership came about and why?
Well, I think if I can start, Janet, the process we went through as a company, as a biotechnology company, is first you want to establish what's called proof of concept. So you usually start with a manufacturing process that gets you quite quickly into the clinic, and where you can show in patients that the product you are developing actually has the effect that you want to see. Now, that is something we established late 22, beginning 23, when we started to see that the acute myeloid leukemia patients we treated showed actually prolonged survival after we had treated them with our product. Then, of course, we needed to think through how we were going to get into a next phase of clinical development, and that also required large-scale manufacturing. And in parallel to our clinical development, we had developed a process which is in principle suitable for large-scale production, but we had not yet identified our what's called GMP, Good Manufacturing Practice, manufacturing partner that requires a specific infrastructure. And we actually also were not sure if we were going to outsource or if we were going to manufacture in-house. Then when we met with NorthX and also with the people behind NorthX, who are now also a shareholder in our company, we realized that this was actually a company with not only an ambition level, but also the capabilities and the factual manufacturing facility that we could use for this large-scale production. And that's how we got started in 2023.
Adding on to what Erik just said, the partnership between our companies was really based on that we could combine our expertise. Mendus had already developed a process, and of course they have the expertise on the therapy. And then NorthX could contribute with the expertise on the GMP manufacturing and having also the facilities that were adjustable so that it would fit the process that Mendus had in mind. So combining expertises from both sides, working together to really establish a facility and all the analytical work in Sweden that Mendus needed, really made a perfect combination, I would say. On top of that, so not only the brick and mortar, but the people really contributed to a successful collaboration, visiting from NorthX to Mendus in the laboratories in Leiden, and also from the Mendus expertise visiting the NorthX facilities. We could really work out an optimal way to work together and establish the facilities as needed for Mendus as quickly as possible.
And so what is the significance of the GMP manufacturing certification?
Well, the significance is that without it, you can't treat people. So you can develop a process as a biotechnology company in your laboratories, which is important because that's where you make it robust, and you can do repeated runs and you can test all kinds of variables to see how robust the process is and how it holds up under different conditions. But at a certain point in time, you have to basically confirm it, lock it in as the process that you want to use to actually treat people in the clinic. But then you also have to transfer it to a GMP facility, and that requires a whole different set of skills and certificates. And I'm sure, Janet, you can say a little bit more about that, because that has been, of course, the reason we shifted to NorthX, which was already a GMP facility.
Yeah, quite often we talk about the valley of death because it's where two different worlds meet. You have the science, the academic, the invention on the one side, and you have the very regulated GMP world on the other side. And as a biotech like Mendus, you've developed a very ingenious way of setting up your production process, but it needs to fit then in the GMP world. And I think it is really smart to team up with people that do that for a living every day. That's really the basis of the collaboration, that you build on each other's strength. And a biotech not necessarily has the strength in GMP manufacturing, but of course they have the invention and they want to make sure that the invention gets to patients. And we share that passion actually, but we work on it from the other side. So we make sure that we use our capabilities, our expertise in manufacturing and analytical control, quality control, to make sure that these inventions reach the patient.
How challenging is that process to achieve?
It's a challenging process because every process is new, but it's also based on the regulations that you have to apply to. So in a way, you build up expertise to bring a new process to the clinic and to market by building on your knowledge from before and tweaking it so it fits this particular process. And again, it's all about collaboration. The colleagues that work at Mendus, they know all about their process. So if we combine what they know about the new process, the new analytical techniques, and what we know about facility adjustments, our quality system, then I think that's a good match to see to it that it becomes successful.
But it is very challenging, Jim, to answer your question from my side. It's been our experience that a tech transfer, as it's called, where you transfer your process from your laboratory facilities to a manufacturing facility, there's never a guarantee. And what often happens is that the manufacturer, which of course also needs to have a certain level of protection, just takes the process the way you have described it in-house and performs it exactly according to the letter of the process the way you have described it. And if it then fails, that's a problem, but it's also something that I wouldn't say it's not their problem, but it's basically, they've carried out your recipe, right? And I think that is where often things go wrong. If you have such a black and white separation of how you transfer a process, then on the one hand, it's, let's say, from a protocol, very clear, but as an outcome, far from guaranteed. And I think here, and that I think made it very unique, also the way we've set it up. It was, on the one hand, a leap of faith from both sides. So I think we really committed to this collaboration, and it was also a joint investment into the manufacturing alliance. So we were really into this from both sides from the beginning. But there was also an incentive to really make it work on a personnel level, and that made the situation quite unique. So I think the way we have quite intensely worked together with also very good team spirit from both sides to transfer the process has first of all contributed to the success of the technology transfer. But it's also been, I think, a very unique and fruitful collaboration.
And I would just say, Erik, for us to be successful as NorthX, it requires that Mendus is successful. We cannot be successful on our own.
Very often CDMOs like, well, it was your recipe, it didn't work, sorry, let's try again. Okay, here's the bill, and it goes on and on and on. And we've been there. I mean, we've had manufacturing failures in the past and what have you, and you have to resolve it. But each time, here's the bill, So, of course, that's how a manufacturer works. But it's just nice if you have a more intimate relationship between two companies that we established, because it works better. It works better in the end.
Exactly. So I don't want to be the CDMO that just performs the recipe. We want to share in the biotech success, and therefore we take a part in that as well.
Right, absolutely. And so where are you at on the clinical trial pathway right now, Erik?
Well, on a very interesting path, Jim, because we started with what's called a phase 1 trial. And in the phase 1 trial, we saw that patients that have a very high level of disease were not doing so well. And that's also now a more common phenomenon that's been discovered in the immunotherapy space. But the positive thing was patients that had a lower level of disease, but still a very high risk of tumor recurrence and in the end death. In the case of AML, it's a very deadly disease, showed a very prolonged survival. So from that phase 1, we learned we have to be in what's called a post-remission phase. So when patients have had their chemotherapy, their disease burden is low, that is where exactly you can train the immune system to take control and prevent residual cancer cells from growing out to full-blown tumors again. That is what we tested in a phase 2 trial. That's what was called a single agent trial. So it was monotherapy, vididencel treatment after patients had achieved a first complete remission following chemotherapy. And that trial went very well. We actually just reported last week at the American Society of Hematology conference in Orlando, Florida, that at 55 months median follow-up, still the majority of patients is alive, with already 8 patients being now followed up past 5 years. So that's a very good outcome in a disease that's normally very deadly. So that sets the stage for us to move into late-stage clinical development in the myeloid blood cancer space. And we will do that in different settings. In acute myeloid leukemia, so there's 2 lines of first-line treatment in AML, and we have an ongoing trial in the post-chemo setting that we also tested in the single-agent trial, but now in combination with an approved AML maintenance drug called oral azacitidine. And we will also test the product in combination with a new first-line treatment, which is called azacitidine plus venetoclax, which is a milder but still very intense chemotherapy. But that is a fast-growing patient population also in need of post-remission therapy. So in the AML space, we are now broadening the addressable patient population, and we have decided to move into chronic myeloid leukemia, which is not so aggressive as AML, and which is also under control with drugs that have been approved, so-called tyrosine kinase inhibitors, and there's a whole range of them. So in principle, the disease is under control, but patients have to take their drugs for the rest of their lives. So here we want to try immunotherapy to see if we can safely have patients stop with their continued drug use and see if the immune system can take over the role of the TKIs. So we are now moving into a phase where we rapidly expand the clinical development of the product vididencel. And of course, for that, it's also very important we have in parallel established the large-scale GMP manufacturing.
And so could you tell me a little bit about how vididencel works, its mode of action?
Yeah, what it is actually is mass-produced leukemic-derived dendritic cells. It's a mouthful, but the long story short is we grow a large amount of leukemic cells from a specific cell line, which is proprietary because you need cells that are stable in cell culture in the manufacturing process. And these cells are not so well recognized by the immune system. That's the known phenomenon, that cancer cells have a way to basically shield themselves from being recognized. So what we do is at the end of the production process, we add cytokines, specific cytokines, which are not normally communication molecules for the immune system. But if we specifically add them to the production process, the leukemic cells start to express what's called dendritic cell co-stimulatory molecules on their cell surface. So that's triggered by the addition of the cytokines. And that's the trick. We irradiate these cells, we store them frozen. So in the end, for patients and for clinicians, it's a very easy to administer product. You thaw the frozen vials and you inject the cells into the skin. And what happens is in the skin, which is a natural barrier to entry, there's a lot of antigen-presenting cells that are basically constantly looking for potential infections or cancer cells, for example. And they phagocytose, so they basically process the product as soon as we inject it into the skin. This is something we have documented in a lot of detail. But the outcome is fascinating. The outcome is that the immune system is trained against a broad range of leukemic antigens, and that does the trick. So we see actually that in most of the patients, the immune system is still relatively good, even after the AML and the chemotherapy. And as soon as the immune system is able to build up an immune response, you start to see these prolonged survivals that we have established in our clinical trials.
And so what makes vididencel different to other immunotherapies?
Well, the main differences are that it's first of all an active immunotherapy. So it means that it trains the immune system of the patient to control the residual disease. That's also the only long-lasting form of immunity. So active immunity, which you also accomplish through, for example, regular vaccination, is something that in principle lasts very long, sometimes even for the rest of your life. Secondly, the product is very safe. So there's a lot of immunotherapies that are administered directly into the bloodstream, where you need all kinds of conditioning regimens before you can safely administer the product. In our case, because you naturally train the immune system, so the antigen-presenting cells in the skin actually trigger a natural immune response, there is no preconditioning or other complicated procedures required. You just inject into the skin. We see very little side effects. We only see some redness in the skin where we inject the product. And then this active immunity builds up that is potentially protective against the disease.
And as far as NorthX is concerned, were there any challenges in adapting to making the product?
I think every process has its challenges and every new project has its challenges. I think we did a really good job together, and the challenges were, of course, timeline to adapt the facility as quickly as possible in the way that was needed for this process, because time here, you say time is money, but here time is important, because the sooner you get to GMP material for large-scale clinical trials, the sooner you can treat patients. So that, I think, is the challenge in every project and in this one too. But together we managed it really well. In a record time, we had adjusted the facilities in the way that was necessary for the Mendus process. So I think we did really well. Then, of course, a challenge to perform the GMP certification and to get the batches all the way to release is often a challenge. But I think none of these challenges are impossible to overcome, as we proved.
We start with the cell line, and the cell line basically already carries all the antigens you need to stimulate the immune system. We don't require genetic engineering or patient material or any complicated procedures in parallel to producing the actual product. And I think that has also helped to make this a scalable process, Jim.
Absolutely, right. And in terms of the facility's capacity to move up in terms of scalability, how would you handle that if it comes to commercial volumes?
Together in this collaboration, we did a scale out of the process that Mendus had developed, and we can certainly scale out further. There's no limitation right now in further scaling out the process in the facilities as we've built them. So I think that is a very positive future prospect for this particular process.
Yeah, so what I think is important, maybe from a, let's say, more technical perspective, we have worked towards a process which is small volume, high density, which means we have a relatively small and compact manufacturing system that can easily be scaled out. So you don't need a huge facility or a room, or it's actually quite a compact production system, but therefore also we don't need very big factories to manufacture this process. And I think also what's relevant is that because we now have large-scale production, we don't need so much for our clinical trials. As long as we continue to manufacture, you also start to what's called stockpile product. And because it's stored frozen and it has a long shelf life, the established shelf of to date is 5 years. So you can already start manufacturing for commercial launch while you are still in the clinic.
It's a good thing to be able to do, absolutely. In terms of the end patient, what are your hopes for those with AML and other similar conditions?
Well, the most important thing is that AML is a deadly disease. And even though there is more and more products, classical chemotherapy and what's called high-intensity chemotherapy, but also now venetoclax, and there's more drugs being developed to reduce the disease to a minimum, but it comes back and it kills patients. So the only curative approach today is what's called a hematopoietic stem cell transplant or a bone marrow transplant, which is a very radical procedure whereby the immune system of the patient is completely destroyed and replaced by a donor immune system. And that comes with 20% transplant-related mortality. It comes with chronic graft versus host disease, where the new immune system basically starts attacking the body in which it has been implanted. That's a very serious condition. So it's been very difficult to control this disease beyond the initial chemotherapy and suppression of the disease. This is where immunotherapy and safe immunotherapy in the form of vididencel can make a real difference, because on the one hand you stimulate long-lasting responses, so durable clinical remissions, but it also comes with a very good safety profile. So the big, of course, purpose would be to try and get as many patients as possible in a long period of durable clinical remissions. And in the chronic myeloid leukemia space, which we are now also entering, it's really focused on also long-term treatment-free remissions. So where you can actually train the immune system to the point that patients no longer need their daily drug use and can live a life free of disease without having to take lifelong medication. So that's the ultimate goal, Jim.
Obviously busy times with clinical trials, but do you have anything else in your pipeline?
Well, we have looked into the use of the cells for other immunotherapies, particularly in solid tumors. We've established initial safety and feasibility in ovarian cancer. We've also looked into whether we can stimulate tumor-infiltrating lymphocytes that we isolate from gynecological cancers, ovarian cancer, endometrial cancer, which can be used for therapeutic purposes. So we've also developed in the background a platform, but it's first things first. Let's first make sure that our mature clinical pipeline comes to fruition, and then we don't have to be bored. We have a deeper pipeline that we can develop into additional indications.
And so how do you see this partnership as contributing to the Swedish biotech ecosystem overall?
For us, a partnership like this is really a good example of the way the ecosystem works nowadays. If we go 10, 15 years back, the larger pharma had a huge role, and they still have a role, but they have a majority role in the ecosystem. What we see now is the biotech, the innovators, are really playing a bigger and bigger role in advancing science to clinic and to market. Sweden has quite a bold ambition to be leading the field of ATMP. And to make that happen, I think these collaborations, like between Mendus and NorthX, are important to let the biotech develop their innovations and combine then forces with manufacturers like us. There aren't that many biotechs that have made it with cell therapy all the way to late clinical phases and have a path forward to the market. So we need definitely more of these collaborations. And I hope that Erik and I can be examples of how the collaboration between Mendus and NorthX really can bring a therapy forward to patients.
Yeah, I can only echo that, Jim. So I think we're on the right track, and hopefully it will create more spin-off and more expertise that will grow out also to the advantage of other companies and as many patients as possible.
And kind of connected to that, what role do collaborations like this play in accelerating innovation for maybe cancers, but beyond cancers? And how do you see this relationship continuing?
I would say it's black and white. You can't do without this kind of collaboration. And like we also described in the practical, let's say, considerations in tech transfer and so on, these things easily go wrong. If you have a high barrier between the developer and the manufacturer, it's a very tricky situation in terms of being able to move these things forward. And I think here we have really accomplished a much more elaborate and intimate relationship between the both companies and the both teams, which has led to success. So I think that is really how it should be done. It also then helps that if you're in each other's vicinity.
For sure. And from our side, we say that we are a CDMO, and that's what we are. But we'd like to say that we are beyond the CDMO, which exemplifies that we're not just performing a recipe that we get from Erik's colleagues. We want definitely to be part of this collaboration in it for the long run. And I think making sure that these collaborations last will benefit the progress of projects like these and very important projects that can make it all the way to patients.
Nice to see the cooperation there between companies, and it's also good to get an insight on how this process works. Hopefully some great results in the future for vididencel and the other products in the Mendus pipeline. So that's it for this week's podcast, and also for January. I'm not really sure where the month has gone, probably in my case trying to stop coughing. I talked to someone in the village yesterday who caught this before Christmas, and she's still suffering. So I've still got lots to look forward to. We're getting into mid-February with interviews already, but if you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. And I will remind you again, as I do every few weeks, because I think it's worth promoting. Unlike some other life science podcasts, we do not charge to be a guest. I like the content to be related to news and what's important. I'm not saying that I don't like advertising, of course I do, but the content on this podcast is 100% independent. And I know there are also other podcasts out there that are exactly the same. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.